Alright, so in this video we're going to be looking at binary fission. Binary fission is a type of asexual reproduction, meaning one cell divides to form two identical cells. This is how prokaryotes multiply. So let's go through the steps together. Step 1, DNA replication. The first thing that happens is the DNA replicates. So instead of one copy, we now have two identical copies of DNA. So make sure you really understand why this is the case. Why do we have to replicate the DNA? Well, we're going to have two daughter cells from this process, and we need each daughter cell to have a copy of the DNA because all cells need DNA. And so if we didn't copy, then the daughter cell wouldn't receive that genetic information, a copy of it. So step 2, the DNA condenses. The DNA then condenses into two circular chromosomes. Think of it like this. Would it be easier to move a messy, tangled string or something really packed? Something really packed, right? The cell condenses the DNA so it can be moved more efficiently during cell division. Step 3, chromosomes attach and the cell elongates. Each chromosome attaches to the cell membrane and the cell begins to elongate. As it stretches, the two DNA copies are pulled apart to the opposite ends of the cell. And this is really important because it ensures that each new cell will then receive one copy of the DNA nicely. So now we've got that cell division. A new membrane and cell wall will form in the middle, and you can think of the chromosomes are just sort of slowly being pulled to the opposite sides and divided now. Now you'll end up with two daughter cells, each containing one identical circular chromosome. Now here's a key idea. This process doesn't just happen once, but it can repeat over and over again. So you can go from one cell to two cells, and then two cells to four cells, and then 4 to 8, you can see it doubles each time, and this is called exponential growth. And this is why bacteria can grow so quickly. For example, when colonies form on a plate, an agar plate, in a really short amount of time, that's because they undergo binary fission. So overall, binary fission is simple but extremely effective, efficient process that allows prokaryotic cells to rapidly increase in number. And that's really important to understand for exams.